Grade 7, welcome back. Let's continue talking about the genetic code, okay? What's meant by a gene? Gene is a section of DNA or a small part of DNA molecule that contains information to code for one specific protein, okay? And D2, a gene is a section of DNA. It's made up of series of nitrogen bases, okay? And these series of bases lie in a row. These bases are arranged in a specific order, like the example in front of you: adenine, thymine, guanine, adenine, cytosine, guanine, cytosine, okay? This is arrangement of series of nitrogen bases, okay, formed a specific gene, okay? And this is help the gene to produce a certain enzyme that activate a chemical reaction to produce a specific type of protein, where the protein can show a specific trait. A single gene on a chromosomes may contain several hundred to million or more of these bases, and each gene responsible for a specific trait is located at a specific site on a chromosome. And due to the presence of many possible combinations between nitrogen bases found in gene, so each individual has a unique set of DNA, okay? So DNA found in all types of body's cell except red blood cells, because red blood cells don't contain nucleus, so they have no DNA. But by taking the sample, blood sample, okay, and examine it, we found that we can obtain a DNA, okay? Because DNA found in a blood samples due to the presence of white blood cells, okay, that having DNA due to the presence of nucleus. So in this lesson, we will talk about order of bases and also the DNA replication. How does gene determine the trait? The order of nitrogen bases along a gene can form a genetic code, and these genetic codes can give enzyme, where the enzyme responsible for activating a chemical reaction to help the gene to produce a specific sequence of amino acids, help to formation a certain type of protein to show a trait, okay? And remember that protein is from organic compound found in the cells of living organisms, and it's a long chain molecule that made of smaller molecules known as amino acids, okay? So by attaching of different amino acids along a chain, this is a arrangement, the specific arrangement of amino acid leads to formation a specific protein, okay? And in genetic code, okay, a group of 3 nitrogen bases can give one specific of amino acid. So, and we also, you should to know, we have 20 and almost 20 amino acids, okay, that can enter the formation of protein, okay, like cytosine, guanine, thymine, okay, can always codes for amino acid alanine. And according to the order of amino acids, the specific protein will be formed and showing a specific hereditary trait, okay? Look at the diagram to can imagine the structure of protein, okay? Or how the protein is formed. This is a different types of amino acids, okay? Each one formed from 3 nitrogen bases, collection of 3 nitrogen bases, okay? We have different types of amino acids, okay? And by linking the different types of amino acid, they will be formed along a chain, Okay, and after formation along a chain of different sequence and different order of amino acid along the chain, the protein will be formed. So a protein is defined as a long chain of a smaller molecule known as amino acids, and the sequence of amino acids will determine the protein's shape and the protein's function. Okay? The last topic, how does DNA copy itself, okay? Or how the DNA replication takes place. Remember the mitosis division, okay? Before the cell mitosis division occurred, okay? The cell prepared itself for division by formation, by making exact copy of genetic material, okay, to ensure that each daughter cell formed from division can receive the genetic information that needs to carry out its activity, okay? So DNA copies itself, and this is known as DNA replication, okay? So DNA replication, the process in which an identical copy of DNA strand is formed, okay, for a new daughter cell. Look at the figure to explain the steps of DNA replication, okay? So we know that the DNA consists of two strands or double strands, okay, and the two strands connected by pairing of different nitrogen bases, okay, and by looking to the nitrogen bases, adenine always paired with a thymine always paired with adenine, cytosine always paired with a guanine, and guanine always pairs with a cytosine, okay? So what happened in the first step? The two sides or the two strands of a DNA molecule unwind, okay, unzips, and separate away between the nitrogen bases, okay? Then the next nitrogen bases that found inside the nucleus begin to direct to the strand and pair up with the bases found on each strand, on each half of the DNA. Look at, this is nitrogen bases found in the nucleus, okay, and this is nitrogen bases begin to direct, okay, the two strands of DNA to can pair with the nitrogen bases found on each strand, okay, and we note that adenine go to the thymine, cytosine go to the guanine to pair with it, okay, and so on. And after this process is end, we obtain the two exact copies of DNA, okay? And by comparing the two exact copies to the first copy, they are the same. At the end of replication, the two identical DNA molecules are formed, okay? So this is known as DNA replication, okay? Thank you, grade 7. Goodbye.